Myself Dipankar Majumdar, and welcome to my channel Microbiologist's Forum. Today we will discuss about the pedigree analysis, and this is the third part, and in this part we will discuss about the sex-linked inheritance. Sex-linked inheritance in man are mainly of three types: X-linked dominant, X-linked recessive, and Y-linked. Genes that may be dominant or recessive carries on sex chromosome, and the sex chromosomes are X and Y. Because of the different distribution of sex chromosomes in males and females produce patterns of inheritance different from genes carried on autosomes. The inheritance of genes on sex chromosomes is known as sex-linked inheritance. Sex-linked means X-linked, since the only defined Y-linked Mendelizing genes are for hairy pinna and the HY antigen. The overall incidence of X-linked disorders are about 0.5 per thousand. Genes carried on X chromosomes are referred to as X-linked, and the genes carried on the chromosomes of Y are referred to as Y-linked or Hollandric genes. Partially X-linked genes are those genes which are carried on the homologous portions of the X and Y chromosomes. Examples of sex-linked or X-linked genes are hemophilia, partial color blindness, deficiencies of vitamin D, etc. And for Y-linked, these are hairy pinna and HY antigen. Now, the X-linked inheritance. A male has only one representative of any X-linked gene. It is always derived from mother, and hence he is said to be hemizygous rather than a homozygous or heterozygous, because its genetic constitution is X and Y. Whether recessive or dominant, an X-linked genes is always expressed in male. A female can be homozygous or heterozygous because the sex-linked chromosomal constitution in female is XX. Since the X chromosomes cannot be transmitted from father to son, a male cannot transmit X-linked trait to his male offspring, that is father to son, is not possible in case of X-linked inheritance. As a father has only one X chromosome which is always transmitted to his daughter, he will always transmit the trait to his daughters in the case of dominant genes, or then carriers in the case of recessive genes. It may be noted that though a female has two X chromosomes, only one member of the pair is active. The second one remains condensed and relatively non-functional and is seen as bar body in the interphase cell. Thus, the females, as in males, there is only one functional X, and in heterozygous females, it is matter of chance whether her paternal or maternal X is in functional state in given cell. This is the example of X-linked recessive inheritance. The classic example of this trait is hemophilia, and here the pedigree chart is shown. Other examples are partial color blindness, that is the inability to distinguish between the shades of red and green, and glucose 6-phosphate dehydrogenase, that is G6PD deficiency, Dachne muscular dystrophy, that is DMD, and probably testicular feminization syndrome. The trait is expressed by all males who inherit the gene. Females are affected only if they are homozygous. Thus, X-linked recessive diseases are often restricted to males and females are uncommon. Mode of inheritance. In hemophilia, the genotypes X, H, Y for affected male and X small h then X capital H for female. X capital H for normal female and X capital H Y for normal male. When an affected male, that is X small h y, marries a normal female, that is X capital H, all his daughters would be carriers, X small h and X capital H, and all his son would be normal. The carrier daughter will transmit hemophilia trait to half their sons because X-linked trait goes from mother to son. When an affected male, that is X small h y, marries a carrier female, that is X small h and X capital H, half the son would be affected and half the daughters would be carriers and half the daughter would be affected. This type of combinations are rare since hemophilia in female is severe and may be lethal. Female hemophilic offspring have a good chance of survival with modern therapy. When a carrier female, that is X small h, X capital H, marries a normal male, that is X capital H Y, half the offspring would be normal, half the sons would be affected and half the daughter would be carrier. And these are the characters of X-linked recessive traits, and two pedigrees are shown here that are for X-linked recessive. An X-linked trait is mostly seen in males, it is uncommon in females. The trait is transmitted from an affected man through all his daughters to half of his grandsons. The trait is never transmitted from male to male, that is father to son. The trait can be transmitted through a series of carrier females. The affected males in a family are related to one another through females. For analysis part, you have to follow the same steps which you have followed for autosomal traits. The first part is to designate the number of generations with Roman numerals, then designate the number of individuals of each generation with English numerals. Then you have to describe the pedigree, how many males and how many females, how many affected male or female, or how many sons, how many daughters, is there any consanguinity or any proband case, all should be described in the description part. Then you have to analyze with the pattern of inheritance that is presented in the pedigree with the help of the characters known for the particular trait. After that, you have to designate the genotypes for each of the individual of each of the generation of the pedigree, so that your pedigree chart analysis will be complete. These are some X-linked recessive traits having a showing with homeme number and phenotypes, etc. Y-linked dominant inheritance. X-linked dominant inheritance. Examples of X-linked dominant traits are vitamin D resistant rickets and XG blood groups. An X-linked dominant trait is seen in homozygous as well as heterozygous females, and in the male having the gene under consideration on his single X chromosome. As a result, X-linked dominant conditions are twice as common in females as in males. Affected male transmits the trait to all his daughters but none of his son. Mode of inheritance: when an affected male marries a normal female, he will transmit that trait to all his daughters but none of his sons. When an affected male marries an unaffected homozygous female, all the children would be affected, the daughter being homozygous. When an affected male marries an affected heterozygous female, all the daughters and half of the son would be affected. When the normal male marries an affected homozygous female, all the children would be affected, the daughters becoming heterozygous. When a normal male marries an an affected heterozygous female, half the children, sons and daughters, would be heterozygous and affected. These are the X-linked dominant disorders. Two pedigrees are shown. In rare X-linked dominant disorders, affected females are twice as common as affected males. Affected males pass on the trait to all their daughters, but none of their sons. Thus, X-linked dominant inheritance can be distinguished from autosomal dominant inheritance only by the progeny of affected males. Transmission by affected females follow the same pattern as autosomal dominant. Affected heterozygous females transmit their conditions to half their children of both sexes. Affected homozygous females transmit the trait to all their children. Now, the Y-linked inheritance, this is also known as Hollandric inheritance. An example of Hollandric inheritance is hairy ears. As the Y chromosome is found only in males, the non-homologous Y-linked genes are found only in males. Males alone get affected, and an affected male transmits the trait to all his sons but none of his daughters. As the Y chromosome is single, genes on Y chromosomes are always expressed phenotypically. Mode of Y-linked inheritance or Hollandric inheritance are: males get only affected, affected males transmit the trait to all his sons but none of his daughters. Here the pedigree of Y-linked inheritance is shown, only the males are affected, and affected males transmit the trait to all the offsprings of him, and the analysis part should be done with the help of the same steps and these are the description of the pedigree, then analysis, then designate the genotypes, etc. That's all. Thank you very much. Thanks for your patience watching.